Good evening. My name is Danielle, and presenting with me is Anuj and Hena. Together we are Team STRIDE, standing for Sub-Second Testing, Research and Investigation of Dopamine and Empathy. Today we will discuss our research regarding the modulation of dopamine and empathy in rats. We will first review some background information, and then we will discuss phases 1 and 2 of our research regarding dopamine and oxytocin and their relationship to empathy. Then we will offer some advice for new teams. So, why are we studying empathy? Empathy is such an important characteristic as it allows for the awareness and understanding of others' feelings. We hope to use our findings in our research with rats in order to translate it to larger scale applications. We are particularly concerned in cognitive empathy, which is the conscious awareness of the mental states of others. This is important when looking into disorders characterized by a lack of empathy, such as autism spectrum disorder, schizophrenia, and antisocial personality disorder. All of these disorders show an abnormal dopamine activity in the brain. As there is limited knowledge in the underlying mechanisms of dopamine and empathetic behavior, treatment in this area suffers, which is where our research comes into play. Now that I've discussed the background on these disorders and empathy, I will now review phase 1 of our research. As dopamine is a neurotransmitter that's involved in the learning and reward processing centers in the brain, we used fast-scan cyclical telemetry in order to measure the dopamine levels in the brain. A probe was inserted into the nucleus accumbens in the rat's brain in order to measure the dopamine levels on a sub-second time interval. The rat model has been shown to be used in neural activity and empathetic behavior research, which is why this is so important for our model. We aim to address the relationship between dopamine and empathetic behaviors using the behavioral patterns in the rats through our paradigms. Our team investigated the relationship between dopamine activity and empathy, as well as how sub-second dopamine release correlates with empathetic behavior. We hypothesized that dopamine levels will have a high correlation with the subjects that express more empathetic behaviors. In terms of our methodology, there were 2 cages. One cage contained the recording rat, while the other one contains the conspecific rat. First, the house light goes on for 5 seconds, indicating that a trial will occur. This is in order to get the rat's attentions. Next, a cue tone will play for 5 seconds, indicating which trial outcome will occur. This can either be reward, punishment, or neutral. Next, the directional light will go on for 5 seconds, indicating which box will receive the outcome. It is shown in this image, the light in the conspecific rat's cage, that the conspecific rat will receive one of the 3 outcomes. Lastly is the delivery of one of the outcomes. This shows shock other, in which the conspecific rat receives the punishment. Shock self is when the recording rat receives the shock. Reward other is when the conspecific rat receives the food pellet, and therefore reward self is when the recording rat receives the food pellet. Lastly, the neutral outcome is when the neutral other is when the conspecific rat receives no outcome, and neutral self is when the recording rat receives no outcome. Our together paradigms have both the recording rat and the conspecific rat. Therefore, each of the 3 outcomes can go to either the recording or conspecific rat. Our alone paradigm trials only contain the recording rat. One of the 3 outcomes can either occur to the recording rat or be delivered to an empty box for our control trial. I will now hand it off to Anuj to discuss our phase 1 results.
Thank you. So, sorry about that. So before we can extract any meaningful conclusions from our data, we first need to make sure that the rats that we trained understand the paradigm they're going through. So on this, on this graph, what you'll see is on the y-axis, percentage of the time that the rats' heads are within their food cage looking for food. And on the top, you'll see each of the cues: house light, trial type, directional cue, and application result. The blue lines are indicative of a reward trial, the green lines are indicative of a neutral trial, and the red lines are indicative of a shock trial. And along the bottom is time in seconds for each of the trials. So, as we can clearly see, the blue lines are the highest, showing that during each of the reward trials, the rats are actively searching their cage for food, showing that they do understand what is going on and they're looking to benefit from it. The neutral trials show just a 0 to 10% relationship of checking, and the shock trials show almost no checking. So once they see that a shock trial has come on, especially after the application of the trial type cue, they never actually check, showing that they at least do understand what each of the trial types mean. Now on the y-axis, you'll see a relative change in dopamine concentration between the axons in the brain, and that's measured in nanomolar from baseline. So this thick blue line is indicative of a reward to self trial, in which the recording rat is given a reward. And as you can clearly see, there's an increase in extracellular dopamine when the trial type comes on and they recognize that it's a reward cue, and once again, the reward is given. Now, it's hard to see, but this thin blue line is the second trial type, in which the reward is still given to the recording rat, but there's no one else in the conspecific cage. The rat is alone. And what's significant here is that with only that difference, the increase in dopamine is actually lower at both of the times that it does increase, showing that the presence of the conspecific increases that dopamine response. It's more positively valence action. This, to us, is indicative of empathetic response. These cyan blue lines show what happens during the trial type in which the reward is given to the other rat, in the case of the thick blue line, and to an empty cage, in the case of the thin cyan blue line. And what we can clearly see is that after the directional cue comes on and the rats that are being recorded understand that the reward is not being given to them, the dopamine decreases because they're not getting a reward. But what's really interesting is that the incidence of dopamine is still higher in the case when a reward is actually given to the other rat as opposed to just an empty cage. This is once again indicative of empathetic response. This thick red line shows what happens during a shock self-trial in which a shock is delivered to the recording rat. Now we can't actually show what happens after the shock is delivered because we're using an electrode within the brain to measure dopamine. And the shock creates too much noise for us to extract any meaningful data from the signal. It just creates a giant spike that goes off the scale. What we can do is show you what happens before that shock. And it's just a steady decrease in dopamine as soon as they recognize that it's a shock trial. Now, the thin red line shows what happens when the shock is still applied to the recording rat, but the recording rat is alone. There's no one in the conspecific. And that decrease in dopamine that accompanies the shock cue is all the more prevalent when the rat is alone. This indicates prosocial modulation, and again, is indicative of empathetic response. Now, when we're searching for empathy, ideally, when another rat is being shocked, we hope to see a decrease in dopamine. And that's actually the exact opposite of what we saw. We saw that in the case in which the shock was delivered to another rat as opposed to an empty cage, dopamine actually increased. And we thought about it, and it does make sense. Because even though we're searching for empathy, you have to acknowledge that there's a self-preservation instinct. And when a shock is being applied to an empty cage, they recognize from the directional cue that they're not getting shocked, but there's still the chance. They still associate that shock cue with pain. And you can't actually tell, either by noise or by visual cue, that a shock to an empty cage is actually a shock. But when they see another rat is being shocked, they have visual confirmation that they will not be punished. And that's why dopamine increases. And these are just our neutral trials used for control, and the only meaningful conclusion we can come from that we can get from this, is that in the presence of another rat, the thick black and green lines, dopamine is higher, just showing increased comfort in the presence of another rat. Ultimately, we can show from our phase 1 data that dopamine is modulated in a prosocial context, and just in general, when a conspecific is present, when the rat is not alone, dopamine levels are higher. And research has shown this is associated with a positive response. And this all shows an empathetic response. Now I'll hand it over to Hanok, who will talk a bit more about our phase 2 data.
Thank you, Nadesh. So in the second phase of our experiment, we wanted to look at the effects of oxytocin on empathetic behaviors. So oxytocin is a neurotransmitter involved in social bonding, and research has shown that oxytocin has therapeutic applications in individuals with mental disorders characterized by a lack of empathy. So our research question is, can oxytocin modulate empathetic behaviors? The team hypothesized that oxytocin will increase empathetic behaviors. Our methodology was very similar to phase 1, except we didn't have any dopamine data with FSCB. Before we placed the rats inside the cage, we had given them oxytocin or saline intranasally 30 minutes prior to the trial. Now that I've given you information on the background, our methodology, and our research question, I'll talk about our preliminary data. So here we have letter presses across 3 different shock trials. The light blue bar graphs, the light shaded bar graphs on the left are indicative of the oxytocin rat groups, and the dark bar graphs on the right are indicative of the rat groups that went through the saline. Now, the blue and red bar graphs are used as controls, and due to time I won't be able to go into them, but we'll focus on the yellow bar graphs here for the conspecifics. Now here we see that there's a significant difference between pressing behavior between the oxytocin and the saline groups. The rats that were administered with oxytocin have a significantly smaller pressing behavior compared to rats that were administered with the saline group. This suggests that our hypothesis is supported because oxytocin modulates empathetic behavior. But what if oxytocin actually modulates the pressing behavior rather than empathetic behavior? So we wanted to see what would happen if we took the conspecifics out. And here we see that pressing behavior between the oxytocin rats and the saline rats have no significant differences. Pressing behaviors in the oxytocin rats actually shot up and are greater than the pressing behaviors of the saline groups, which this data further supports our hypothesis that oxytocin does in fact modulate empathetic behavior. So our overall conclusions are that our phase 1 data supports our hypothesis of the modulation of dopamine in a social context. And our phase 2 preliminary data is very promising, and by next year we will have data to submit to journals and conferences. So here are some takeaways for the new gemstone team. Challenges that all gemstone teams will face include scheduling, times to meet, and communicating inside and outside of your meetings. So make sure that you get started early, and make sure that you know exactly what you're going to be doing so you can gather some data for your research. One thing to take into consideration when you're making a gemstone team is to have a broad range of majors, like we do up here. And one of the benefits of having a range of majors is it brings new perspective into your research, which will benefit your research as a whole. Another thing is to stay on top of your work and make sure you get everything done early. And at the end of your meetings, make sure you assign responsibilities to each of your team members and have team roles assigned so that your next meeting will run smoothly. We'd like to acknowledge all of these people here for helping us with our gemstone research, especially our mentor, Dr. Matthew Rush, and our lab assistant, Brian. We have come to the end of our presentation. We'll open the floor to some questions, including gemstone freshman back there.